Lecture 24, Contraction and Relaxation of the Muscle Fiber. Contraction and Relaxation of the Skeletal Muscle Fiber. During this lecture, we are going to study the nervation of the skeletal muscle fiber and the mechanism of contraction and relaxation of the muscle fiber. We will start with the introduction to the neuromuscular junction. Introduction to the neuromuscular junction. Skeletal muscles are innervated by somatic efferent neurons. And stimulation of these anterior horn cells in the spinal cord produce nerve impulses. These nerve impulses will be conducted through A-alpha nerve fibers to the muscle to produce skeletal muscle contraction. And the junction between the nerve terminal and the muscle surface is known as the neuromuscular junction, or it is a type of a chemical synapse. It is a type of a chemical synapse where there is a contact between the nerve terminal and the muscle surface. The synapse is formed of a presynaptic neuron and a postsynaptic surface. The presynaptic neuron is the termination of the A-alpha motor nerves, and the postsynaptic surface is what is the surface of the skeletal muscle that contain the receptors. The presynaptic neurons are large myelinated nerve fibers of the A-alpha motor neurons. Their cell bodies are located in the spinal cord in somatic nerves or in the brainstem for cranial nerves. Their cell bodies are located in the spinal cord or in the brainstem. Usually, each muscle fiber has only one neuromuscular junction. Terminal nerve endings makes a junction at the midpoint of the muscle fiber. What is the meaning of the motor unit? Motor unit is composed of one motor neuron and all the muscle fiber that it innervates. One motor neuron and the muscle fiber that it innervates. So the motor unit means the motor neuron, the somatic motor neuron, plus the skeletal muscle fiber innervated by it. Muscle fibers in a single motor unit are located in one muscle but scattered throughout the muscle. This means that this motor unit is supplying different number of muscle fibers. They are scattered in the muscle, but all are located in one muscle. A single fiber is controlled by only one motor unit. A single fiber is controlled by only one motor unit. The number of fibers innervated by the single motor neuron varies from few to thousands. This means that we have different types of muscles inside our body. Muscles that produce skilled movement, like the muscles of the larynx. Muscles that produce movement of the eyeball. Muscles that control the skilled movement of writing or typing. In this situation, the motor unit is going to supply limited number of muscle fibers. Few muscle fibers. In the larynx, the motor unit is going to supply 2 to 3 fibers. Muscle controlling the movement of the eyeball is supplying 10 to 20 fibers, while in muscles that produce gross movements like the biceps, like the gastrocnemius, in this situation, the motor unit is going to supply huge number of muscle fibers. When an action potential occurs in a motor neuron, all the muscle fiber in its motor is going to contract. neuromuscular junction. That is the shape of the neuromuscular junction. It is formed of the presynaptic nerve terminals, which is known as the synaptic knobs or the synaptic buttons. These are nerve terminals of the somatic efferent containing vesicles that contain thousands of acetylcholine molecules, contains mitochondria for ATP synthesis, and their membranes contain specific type of voltage-gated calcium channels. So, when the axon reaches its terminal, it loses its myelin sheath and it has a dilated end called synaptic knob or synaptic button. This synaptic knob or synaptic buttons containing vesicles that contain thousands of acetylcholine molecules, contains mitochondria for ATP synthesis, and also the nerve terminal contains specific type for voltage-gated calcium channels. Synaptic buttons or synaptic bulbs contain synaptic vesicles which contain molecules of the neurotransmitter which is acetylcholine. What are the events that occur in the neuromuscular junction? What is the mechanism of transmission of the nerve impulse from the nerve to the skeletal muscle? The action potential reaches to the nerve terminal, and when the action potential or the nerve impulse reaches to the nerve terminal, it stimulates the opening of the voltage-gated calcium channels. This will trigger calcium influx, and calcium is going to bind with the vesicles, causing them to move, fuse, and rupture to release the acetylcholine. So, when the nerve impulse reaches to the nerve terminal, it stimulates the opening of the voltage-gated calcium channels, producing calcium influx. Calcium binds with the vesicles, causing them to move, fuse, rupture, and releasing the acetylcholine to the synaptic cleft. So, again, what is the changes occurring in the nerve terminal? Opening of the voltage-gated calcium channels stimulates calcium influx. Calcium stimulates the rupture of the acetylcholine vesicles and release of the acetylcholine into the synaptic cleft. Acetylcholine cross the synaptic cleft, go to bind with a specific type of nicotinic cholinergic receptors. Nicotinic cholinergic receptors. And these receptors produce opening of sodium channels, the ligand-gated sodium channels, producing sodium influx and potassium efflux. Sodium influx and potassium efflux, and the sodium influx is 7 times the potassium efflux. So, at the end, we will get depolarization of the postsynaptic membrane of the sarcolemma due to sodium influx that is more than potassium efflux. We have depolarization of the postsynaptic membrane or the sarcolemma. The area of the sarcolemma where the neuromuscular transmission occurs is called end plate, and the depolarization is called end plate potential. The state of depolarization that's located in this part of the sarcolemma is known as the end plate potential. The change in the membrane potential of this area is caused by sodium influx, and the sodium influx is greater than the potassium influx, so the result is depolarization of this part of the membrane, and this occurs due to binding of the acetylcholine with the cholinergic nicotinic receptors. Acetylcholine comes from the nerve terminal in response to the entry of calcium through the voltage-gated calcium channels, and this occurs in response to the spread of depolarization to the nerve terminal, stimulating again calcium entry, stimulating release of acetylcholine binding with receptors, and depolarization of the sarcolemma, and this is known as individuation. Functional anatomy of the neuromuscular junction. If we look to the neuromuscular junction, neurotransmitter vesicles release acetylcholine at a point called active zone. Active zone is a part present in the presynaptic membrane opposite to the junctional folds of the sarcolemma. So, the part at which acetylcholine is going to be released from the presynaptic neuron is known as the active zone. Junctional folds increase both synaptic surface area. This junctional folds increasing the post-synaptic membrane surface area, and it is located for most of the acetylcholine receptors. We mentioned it previously as the cholinergic nicotinic receptors. It is ligand gated for both sodium and potassium channels. Basal lamina and both synaptic membrane contain many molecules of an enzyme known as acetylcholine esterase enzyme. Acetylcholine esterase enzyme. This enzyme produces breakdown of acetylcholine. So, after the acetylcholine come from the nerve terminal, bind to the receptor, end its action, what's going? It's going to be hydrolyzed by the enzyme acetylcholine esterase into acetic acid and choline. Most of the choline will be reuptaken again by the nerve terminal. Most of the choline coming by the hydrolysis of acetylcholine by the enzyme, the choline is going to be uptaken by the nerve terminal to be used for the resynthesis of acetylcholine. This arrangement increases the chance of acetylcholine to bind to its receptors. Summary of the events at the neuromuscular junction. Number 1. Activation of acetylcholine receptor. Two molecules of acetylcholine bind to a receptor. Opening of the acetylcholine receptor ligand-gated ion channels cause sodium to flow across the membrane, and this is the action of acetylcholine through the cholinergic nicotinic receptors. Production of the muscle action potential. Generation of end plate potential, and when this end plate potential reaches to the firing level, it's going to generate muscle action potential. And the muscle action potential is going to spread along the muscle surface, along the sarcolemma, until it reaches to the T-tubules. Then, termination of acetylcholine activity, hydrolysis of the acetylcholine by the enzyme acetylcholine esterase, and hydrolyzing it into acetic acid and choline. Choline is going to be reuptaken into the presynaptic membrane and reused to synthesize acetylcholine by the enzyme acetylcholine synthase or choline acetylase enzyme. So, action of acetylcholine is going to be terminated. Acetylcholine is not allowed to bind permanently with the receptors. Acetylcholine is not allowed to bind permanently with the receptors, because permanent binding of acetylcholine to the receptor will produce some sort of a block, some sort of a depolarizing blocking of the receptor. So acetylcholine binding to the receptor, opening the sodium channels, allowing the generation of the end plate potential, and this end plate potential going to spread or change into an action potential after reaching to the firing level, spreads along the surface of the muscle, and after that what's going, the action potential is going to be what? Is going to be spread through the T-tubules to the deep muscle fibers, and acetylcholine action should be terminated. The termination of the action of acetylcholine by the enzyme, which we known as acetylcholine esterase enzyme, and this acetylcholine esterase enzyme hydrolyzes the acetylcholine into acetic acid and choline. Choline will be reuptaken by the nerve terminal to be reused for the synthesis of then acetylcholine by acetylcholine synthase enzyme. This process repeats itself as long as there is a nerve impulse arising at the neuromuscular junction. Electrical activity at the neuromuscular junction. That is the alpha motor neuron supplying the skeletal muscle and releasing of the acetylcholine in response to the action potential generated in the nerve fiber. So that is the shape of the action potential generated in the nerve fiber. And in response to this action potential generated in the nerve fiber, we have action potential generated in the skeletal muscle. It is formed of also depolarization and repolarization. And in response to the spread of this muscle action potential through what? Through the sarcolemma and to the deep muscle fibers through the T-tubules, we have generation of something which is called the muscle contraction and relaxation, the simple muscle twitch generated by the muscle in response to stimulation. And as we will mention next, the phases of the simple muscle twitch. So that is the electrical changes occurring in the nerve fiber. That is the electrical changes occurring in the muscle fiber, and that is the mechanical changes appearing in the muscle fiber due to the electrical or the changes in the action potential generated in the skeletal muscle fiber. Neuromuscular junction has something called a safety factor. What's the meaning of a safety factor? It releases sufficient neurotransmitters. It releases huge amount of acetylcholine in order to what? To generate the muscle action potential. Usually 1,000 or so times over. 1,000 or so times over. So huge amount of acetylcholine are released by the nerve terminal to ensure the development of the muscle action potential in order to ensure the production of the muscle response. Some application, we have myasthenia gravis. It is an autoimmune disease characterized by the formation of antibodies against the cholinergic receptor. It's characterized by muscle weakness. It produces muscle weakness. Muscular dystrophy. Muscular dystrophy is abnormality or mutation in the dystrophin protein which brings the tension created in the sarcomere and transmit it to the sarcolemma of the muscle fiber. Rare competitive neuromuscular blocker used to produce muscle before surgical operation. Botulinum toxin or the botulism toxin is a certain type of toxin produced by a bacteria which is a Clostridia botulinii. This type of bacteria usually grow on what? Grow on the beef or meat, the canned beef or fish or meat, and in this situation, the poisoning by the Clostridium toxin, by this bacterial toxin or by the botulinum toxin, inhibit the release of acetylcholine. So it may lead to death due to paralysis of the diaphragm and external intercostal muscles. Nerve gas. It inhibits the acetylcholine esterase permanently. And this will produce permanent binding of acetylcholine to the receptors, and this will produce muscle paralysis. Events of excitation-contraction coupling in skeletal muscle fiber. Excitation-contraction coupling is the event that link the electrical activity to the skeletal muscle contraction. As we mentioned, the depolarization wave will spread along the sarcolemma and the T-tubules. This will stimulate the release of calcium from the sarcoplasmic reticulum. Calcium will interact with troponin C, moving troponin and tropomyosin away from the binding sites. Allow the head of myosin to interact with the actin binding sites, and when this occurs, there is a sliding of actin over myosin and the production of contraction. So that is the relaxation. when there is no binding of troponin with calcium, and binding of troponin with calcium moving the tropomyosin away from the binding sites, allowing the sliding of the actin over myosin. If we look for the summary of the excitation-contraction coupling, muscle action potential propagates along the sarcolemma and the titubules. Depolarization of the titubules was going to activate the dihydropyridine receptors. Activation of the ryanodine receptors on the sarcoplasmic reticulum. Calcium flow out from the sarcoplasmic reticulum, increasing the calcium concentration in the cytoplasm. So we now change the electrical events, which is a depolarization wave that spread along the sarcolemma and the titubules, that leads to the release of the calcium. And once the calcium is released, we are going to start the mechanism of skeletal muscle contraction. At the end of the action potential, calcium release channel close, so no more calcium comes from the sarcoplasmic reticulum, and we are going to pump the calcium back to the sarcoplasmic reticulum by the calcium ATPase. Calcium will bind with the calsequestrin protein, inside the sarcoplasmic reticulum again. So that is with the end of the action potential. Role of calcium in cross-bridge cycling or cross-bridge formation. We mentioned calcium is stored in the sarcoplasmic reticulum. Release of the calcium. Calcium is going to bind with what? With the troponin C. We have here the head of myosin, and the head of myosin would like to bind with the actin binding sites. These actin binding sites are covered with the tropomyosin, so that is the relaxed state or relaxed situation. With the binding of calcium with the troponin, the tropomyosin will be moved away from the binding sites on the actin, allowing the sliding of actin over myosin and the generation of muscle contraction. The contraction cycle, the sequence of events that cause the myosin filament to slide. Number 1, nerve impulse arrive to the nerve terminal, releasing the acetylcholine. Acetylcholine diffuse and bind with the receptor, generating the motor end-plate potential that can spread, generates depolarization that spread along the muscle surface, sarcolemma and the tubule, and the acetylcholine stays in the synaptic cleft, destroys acetylcholine, so another muscle action potential does not arise unless more acetylcholine come from the nerve terminal. Then the muscle action potential triggers the release of calcium from the sarcoplasmic reticulum. Calcium go bind with the troponin, and this will move the tropomyosin away from the binding sites, sliding of actin over myosin and producing contraction and shortening of the sarcomere. That is the sarcomere in the relaxed situation, that is the sarcomere in the contraction phase. Then we have, after the end of this action potential, what's going? Calcium is going to be pumped back inside the sarcoplasmic reticulum. Calcium is going to be pumped back inside the sarcoplasmic reticulum, and then this calcium bind or pumping to the sarcoplasmic reticulum allows the tropomyosin to come back and cover the binding sites to produce muscle relaxation. So the events start with the action potential delivery to the nerve terminal with release of the neurotransmitters, acetylcholine, binding with the receptor, generating of the end-plate potential, generation of depolarization. Acetylcholine is going to be hydrolyzed by the cholinesterase to terminate this action potential of the muscle until another acetylcholine is released by another nerve impulse. Release of calcium from the sarcoplasmic reticulum due to activation of dihydropyridine receptors and ryanodine receptors. Calcium bind with troponin. Troponin moves tropomyosin away from the binding sites. Contraction of the sarcomere. Bumping of calcium back to the sarcoplasmic reticulum. Return of tropomyosin to its original position. Muscle relaxation. the model of cross-bridge formation. That is the head of myosin, and the head of myosin would like to bind with the actin binding sites. The head of myosin has an ATPase activity and hydrolyzes this ATP into ATP and phosphate. It is in a detached state. When it releases the phosphate, it's going to produce binding with the actin binding sites. It's going to show binding, attachment. Then, with the release of ADP, it's going to produce bending. It's going to produce bending or the movement of the actin filament towards the center of the sarcomere, generating the power stroke. Then, it has to be detached and return to the original position. And this will occur when it binds with a complete molecule of ATP. When it binds with a complete molecule of ATP, it will be detached, and this ATP will be hydrolyzed again into what? Into ADP and phosphate. Release of phosphate produce binding. Release of ADP produce bending, and complete binding with one ATP molecule. What is going? There is detachment of the head of myosin from the binding sites on actin. Contraction is produced by the sliding of actin over the myosin or the thin filament over the thick filament. That is the shape of the head of myosin, and the head of myosin is going to bind each time with actin binding sites, bringing the actin filament towards the center of the sarcomere, and this is occurring when the turbomyosin moves away from the binding sites, and this usually occurs when calcium, which is this yellow dots or yellow balls, bind with the turbomyosin, moving the turbomyosin away, and this condition of the generation of power stroke by the heads of. So, summary of the contraction cycle. ATP hydrolysis on the myosin head. ATP is hydrolyzed by ADP plus inorganic phosphate. This energized the myosin head. ADP plus inorganic phosphate remain attached to the myosin head. Attachment of myosin to actin to form cross bridges. This energized myosin head attaches to myosin binding sites on actin and the release of the inorganic phosphate. The generation of power stroke occurs with the release of ADP. Then the myosin head will be detached when it completely bind with a complete molecule of ATP. Both contraction and relaxation are active as both of them require ATP. Splitting of ATP by myosin ATPase provides energy for power stroke of cross bridges. Binding of fresh molecule of ATP to myosin lets the bridges detach from the actin filament at the end of power stroke, so cycle can be repeated. Active transport of calcium back into the sarcoplasmic reticulum during relaxation depend on the energy derived from the breakdown of ATP. So, both contraction and relaxation needs ATP. ATP is needed for the cross-bridge formation. With the release of phosphate, we have binding. With the release of ADP, we have the bending. Then detachment needs another one molecule of ATP to bind with the myosin heads. Pumping of calcium inside the sarcoplasmic reticulum also needs ATP. Rigor mortis or rigidity of death. After death, the cellular membranes become leaky to increase the calcium in the sarcoplasm. So we have increased calcium, and we have stop of the mitochondria to work. We have decrease in the ATP formation, so ATP synthesis decreased. So we have calcium in the cytoplasm or in the sarcoplasm, and we have decrease in the ATP. So what's going? The muscle is going to show rigidity. What's the meaning of rigidity? It is the state of contraction or state of maintained contraction of the muscle. Myosin head cannot detach from the actin due to absence of complete ATP molecules, and the head of myosin become no pumping of calcium to the sarcoplasmic reticulum due to absence of ATP, so the muscle is going into continuous contraction for about 24 hours, and this usually begins 3-4 hours after this. Then the enzymes is going to produce autolysis to digest the cross bridges. Twitch contraction. It is the brief contraction followed by relaxation due to stimulation of the muscle by maximal stimulus. Single maximal stimulus of the muscle is going to produce contraction followed by relaxation. The duration of the muscle twitch varies from 20 milliseconds to 200 milliseconds according to the muscle type. The phases of the simple muscle twitch or the muscle twitch include latent period, which is a period between stimulation and appearance of response. During this, we have generation of the action potential in the muscle fiber, spread of the action potential across the tubule, and the release of calcium from the sarcoplasmic reticulum. Then we have the second phase, which is the contraction phase, which ranges from 10 to 100 milliseconds. Cause or the changes that occur during the contraction of the binding of the calcium with troponin, then myosin binding sites on actin, and the cross bridge formation. The last phase is the relaxation phase, caused by the active pumping of calcium inside the sarcoplasmic reticulum. Myosin head detached from the binding sites, and the muscle is going to increase in length and the loss of shortening. Myosin head detached from actin. Muscle fiber that move the eye, the muscles of the extraocular muscles or the muscles of the eye, have a contraction period lasts about 10 milliseconds. They are fast muscle fibers. They produce fast contraction and fast relaxation. Contraction refractory period. We have a condition in the cardiac muscle which is no tetanization of the cardiac muscle, while the skeletal muscle, as we are going to see in the next lecture, can produce a tetanus contraction. Why? Skeletal muscle can produce tetanus, which is a continuous contraction without relaxation, while the cardiac muscle cannot produce tetanization. And if you remember that during the contraction of the cardiac muscle, we have a refractory period called absolute refractory period. That is the action potential of the cardiac muscle or the ventricular muscle fiber. We have a plateau, and we have here systole and diastole. During systole, no stimulation of the muscle can produce another contraction, or the cardiac muscle cannot respond to any stimulus during the contraction. Why? Because of the presence of the refractory period. The refractory period in skeletal muscle is 5 milliseconds. So the muscle can be stimulated during the contraction, while the refractory period in the cardiac muscle is 300 milliseconds, so it cannot be stimulated during. That is the shape of two sarcomeres before contraction in a relaxed muscle, with partial contraction, with maximal contraction. What is the change? You see the shortening of the sarcomeres. Why there is shortening? Because of the sliding of the thin filaments over the thick filaments by the cross bridge cycling, by the process of binding, bending, detachment. Okay, that is the I band, that is the A band. If we go and look, what are the changes that appear in the sarcomere? The I band decrease, the H zone decrease, the whole sarcomere decrease, but the A band is kept constant. So during sarcomere contraction or during skeletal muscle contraction, sarcomere shorten, I band shorten, H zone shorten, while the A band is not affected. It is apparent here in the electron microscopy picture. We have here the A band, and in the center we have H zone, and this is the I band. That is the line, so that's I band, and that is I band. With muscle shortening or sarcomere shortening, the sarcomere shorten, the I band shorten, the H zone shorten, the A band is kept constant. Lens tension relationship. What is the meaning of the lens tension relationship? We have a curve that studies the relation between the length of the sarcomere and the tension created in the muscle. That is the resting sarcomere length, percentage of optimum. That is the tension developed during contraction, percent of maximum. As the length of the sarcomere increased gradually, the tension created in the muscle increased. Until what? Until we reach to a certain point where the tension becomes maximum, and any further increase in the length of the sarcomere associated with decrease in the tension of the muscle. So why we have here the maximum tension with the optimum length of the sarcomere 2.2 micrometer, we have here the maximum binding between actin and myosin, the maximum binding between head of myosin and binding sites of actin. As we change the sarcomere length by decrease or by increase in the sarcomere length, the tension go down, the tension decrease. Why? You decrease the binding between actin and myosin. You decrease the binding between actin and myosin. That is the length tension relationship. Study the relation between the length of the sarcomere and the tension developed inside the muscle. The relation is not fixed. When the length increase, the tension increase until it become fixed, then go down. Why? The optimum length of the sarcomere, 2.2 micrometer, produce the maximum binding between head of myosin and actin binding sites. If you decrease the length of the sarcomere, you decrease the binding. If you increase the length of the sarcomere, you decrease the binding.